Okay, good afternoon everyone. I'm Jishen Liu from the Faculty of Bioscience Engineering, and today I'm going to talk about an article we have published at the beginning of this month in the journal PLOS ONE, and I'm going to talk about the work in this article. It's about the search for limiting factors in the RNA pathway in the RNA binding protein R2D2. So first, let's get to know what is RNA interference. It is short for RNAi, and it has won the Nobel Prize in Medicine in 2006, and it's a very important technology in the RNA biology. So the RNAi process will be achieved through a specific gene sequence by a group of small RNAs, and in the insects, the in vivo application for this technique is through the injection of the double-stranded RNA to the hemolymph, and it will cause the systematic RNAi. So this is a figure of the very basic RNA process. So first, the long double-stranded RNA will be cut into small duplex by the enzyme dicer. And then one of the strands of this duplex will bind to the apoprotein, and they will form a complex. And then this complex will actively search for the target messenger RNA here. And then it will degrade the RNA to achieve the gene sequence. So in this process, this RNA binding protein R2D2, which is reported to be could stimulate the RNA process in the Drosophila, the fruit fly. So it is interesting to us, and for the following work, they are based on this protein. So I'm studying the Suchomonas moran. It has been domesticated for thousands of years and can produce the silk. And why do I choose this insect to study? Because it is the model insect in the Lepidoptera insects. And for the RNA mechanism in this insect, it is not clear to us, because the genomic map is clear for this insect, but the RNA machineries remains unknown. So it's very important to study to get increased knowledge of the RNA factors in this kind of insect. So these are the materials and methods I use in the research, including the messenger RNA expression study, generation of double-stranded RNA, cell line transfection, and RNA functional analysis, Western blot, immunofluorescence microscopy, and the sequence comparison. And these are some of the results we have obtained here. So first, I've investigated different kinds of factors to study their expression pattern in the larval tissues and in the pupal tissues, and also in the BM5 cells. It's a kind of cells from the moms, the Suchomonas. And here, the actin gene is used as an internal control. And we can see for most of the factors here, they have a very broad expression pattern in the larval tissues and also in the pupal tissues. And one of the factors which is interesting to us is the R2D2 protein. We could only examine the expression in very limited tissues, like the central nervous system, the testes, and the ovarian. And also in the pupal tissues, a clear expression can be observed after 30 cycles of PCR here. So, does this limitation expression of this factor can contribute to the RNA process? So, we have do the following experiment. So first, we use different kinds of specific double-stranded RNA here at different concentration to check if there's a reduction of the activity of this loop luciferase gene. And you can see here, compared to the control, there's a reduction of the activity. So it means RNA process occurs here, and it's also a dose-dependent reduction. So, because as I talked before, the R2D2 is a binding protein, and it was reported in the Drosophila can activate the RNA process. So, if we introduce the R2D2 here, this RNA process can be also activated again. So, because we could not clone the open reading frame from the mommy's neuron, so we transferred the RBD2 gene from the tribodium because they had two similar double-stranded binding domains, which are underlined here. And then, after the introduction of the tribodium RBD2, you could see there's still a reduction, a dose-dependent reduction here. However, when you compare to the one without the introduction of the tribodium here, you see when in the same concentration, there's no very obvious reduction. So it means maybe the cells let some kind of component to be activated by the double-stranded RNA. So to solve this problem, we decided to generate cell lines which could express the tribodium RBD2 endogenously. And then we test the cells with DNA, non-specific double-stranded RNA, and specific double-stranded RNA at different concentration. However, when compared to the control, there's no obvious reduction of activity. So it means the RNA process is not happening here. So one possible explanation may be because the cells, the tribodium RBD2 is ectopy expression here. So this might be the reason we could not detect the RNA response. So it is also reported the induced expression can activate the RNA response. So we use the double-stranded GFP because it's the induced expression gene. And you can see here, just compare the one, the double-stranded GFP, this one, and this one. You can see there's a reduction. So it means there's RNA happening here. So next, we want to know the localization and accumulation of the tribodium RBD2 in the cells. So we isolated the proteins and we used the Western blot to detect the accumulation of this protein. And you can see here the tribodium RBD2 protein. It locates more in the cerebral fraction of the cells than the incerebral fractions of the cells. And when we see the fluorescence microscopy, we use the green fluorescence to detect the presence of this protein. You can see there's some small green lines here, which means the presence of this protein. And it is located not for the overview of the whole cells, it's just some defined areas. So it might, because this kind of protein is also related to the RNA localization. So in conclusion, the RBD protein is limited expressed in the chicken mouse tissues, and RNA occurs even without the mommy's RBD2 in the real-life cells, because maybe other factors can compensate the absence of the mommy's RBD2, and we also isolated some mutant alleles. And the ectopy expression of the tribodium RBD2 could not help to activate the gene silence. Maybe because it's ectopy expression, and for this kind of protein, it locates in some defined areas of the cells. Anyhow, our research provides a first approach to study the ion response in the particular insects. So that's all. Thank you.
What's the logic next step?
The next step, maybe we try to clone the whole frame from the mommy's mori, not from other insects. Mm-hmm. So maybe if we can successfully clone this gene, then we can test them in the cells again. So then we can test maybe this protein is helpful or not helpful to the RNA response.
In practice, once we have that system down, what can we do with it in practice?
I think this is a kind of theory study because this is a factor involved in this process, and in fact this process is very complicated. So maybe other factors can compensate, or other factors will, other factors will, maybe the function because of other factors, but not these factors, so we have to examine first. And after that, if we have confirmed the function of this protein for the RNA process, then we use it in the practice.
And is application then possibly very wide? I'm thinking of, I've read about the Nature Methods, the journal, describing how you can use special proteins that react to light and that you can actually consider them a switch to switch on certain cells and bodies. Do you think that this could lead to similar kind of switching on to act as a switch for certain cells, or is that not an option?
So you mean because we switch this one to the cells?
Well, what they found is that there were certain proteins, and when you emit light, and then they react to the light by doing a certain reaction. I think it was in green jellyfish or so, Nobel Prize in Chemistry, I think 2007, and they tried to take that kind of system, and they said, well, if we can take that protein, and if we can, through a virus, insert it, for instance, in the brain, we can make certain cells fire or switch them off. And in that way, they really created a biological switch, simply by adding lights, on, off, on, off. Well, I wonder, I think it's the same procedure. Yeah. Because that technique is being applied in many different applications, so I think that.
So it will just block the Bombyx Moris RNA. It will just functionless. It will not work anymore.
So it's also kind of switch that you make.
Switch
off the function of this, maybe a kind of specific gene or specific protein. Yes, we can just block the expression of this function.
Promising.
Yeah, it's really promising. This RNA interference, really.
I think it's quite big.
We are also doing some research on our group also doing some kind of research. We are doing that RNAi. They are doing in Bombyx mori. We are doing in nematode. So when they will infect the plant, the nematode will inject some RNA from the plant, and will insert some RNA artificially to the nematode, and it will just make useless and functionless.
It can functions.
Yeah.
One of the things about, probably it's the same technique, the one that I talked about, is that you can see nematodes functioning, and just by shining light on it, they completely cease to function. They completely halt moving, and then you switch the light off again, and they move again. You switch the light on, and they freeze. It's fascinating work. Congratulations. Thank you very much.